Start. Hello everyone, my name is Jayme James, and today I'd like to introduce my research topic in which we discovered that apigenin was able to induce apoptosis in MDA-MB231 cells in vitro. To give a little overview of my presentation, I'll first get into an introduction where I'll introduce everyone to important concepts and compounds that are pertinent to this research project. We'll also look into some objectives in which we'll discuss what we were really trying to achieve throughout this process, as well as look into our experimental methods, primarily the MTT assay, the caspase-3 assay, and the flow cytometry test. Additionally, we also conducted another assay, MTT assay, on a different cell line called HEK293 T cells. We'll end with our conclusions and also look to some future directions. I'm sure most people in this room have either personally known someone or has at least heard of someone that has been diagnosed with breast cancer. This is because breast cancer is one of the most commonly diagnosed cancers in the world. According to the CDC, among women, there are around 264,000 new cases of breast cancer each year. This means that out of all of the cancer cases for women, breast cancer makes up 29% alone. It is the second most common type of cancer to impact women, coming second only to skin cancer. To put this into perspective, if you were to take all of the women in the United States and place them in this room, 1 in every 12 individuals would be diagnosed with breast cancer at least once in their lifetime. Due to the prevalence of breast cancer, it is very important that we find effective treatment methods to combat this specific disease. So in our research project, we looked at a compound called apigenin. Now, even if you have never heard of apigenin before, I'm almost positive that everyone in this room has ingested it at least once in their lifetime. This is because apigenin can be found in a lot of commonly known plant sources that we can often pick up at our local grocery stores. For example, parsley, celery, spinach all contain apigenin. Some less known sources are chamomile and passion flower. Now, apigenin is part of a subclass of compounds known as flavonoids. Flavonoids are found in many plants and offer a lot of protective services to the plants themselves. It can help protect the plants from sunlight, protect them from predators, as well as even attract pollinators. In humans, it's known to have a lot of therapeutic benefits, including acting as an antioxidant and an anti-inflammatory agent. Looking at knowing apigenin's therapeutic benefits, it has actually been studied on a variety of different cancer cell lines. For example, in colorectal cells, a study showed that apigenin was effectively able to decrease cell proliferation in these cells, as well as in lymphoma, where it was able to increase the production of p53, and in melanoma cells, it was able to help our immune system's sensitivity to cancer cells. In our research project, we were specifically looking at MDA-MB231 cells. Now, these are a highly invasive and aggressive triple-negative breast cancer cell line. Now, the triple-negative means it lacks the estrogen receptor, the progesterone receptor, and the human epidermal growth factor. When a breast cancer lacks these 3 receptors, it means that it cannot be treated by hormone therapy, and the primary treatment methods are usually chemotherapy or surgical methods, which we all know is very harmful to the body. Therefore, we set out with the 3 primary objectives. Initially, we just wanted to see the effect that apigenin would have on the proliferation of these breast cancer cells. We hypothesized that as the concentration would increase, the proliferation of breast cancer cells would decrease. We also wanted to see the effect that apigenin would have on our immune systems. So we specifically looked at a protein called PD-L1. Now, I'll explain a little bit more what this protein is later on. However, for now, we hypothesized that as the concentration of apigenin would increase, the expression of PD-L1 would decrease. Furthermore, we wanted to see what effect apigenin would have on the normal cells in our bodies, and we hypothesized that the different concentrations of apigenin would have no effect on the normal cell lines. So to start off with our experiment, we tested 5 different treatment groups. The 10 micromolar treatment of apigenin, 20 micromolar, 40 micromolar, 60 micromolar, and 80 micromolar. We also had a control group which had no treatment of apigenin and just contained the MDA-MB231 cells. Now, if you look to the picture on the left, if you look closely, you'll notice that these cells have a very elongated, irregular epithelial-like nature to them. However, as the concentration of apigenin increases, they become more circularized. They also, it starts to form clumps together. And these are still images, so it's not really visible here, but when we looked at it through the microscope, we could see a lot of floating cells, which is all indicative of cellular death. Furthermore, there are, in the two highest concentrations, there are a lot of black spots, which is indicating cellular debris. So therefore, just with our microscopic results, we were able to determine that apigenin did have some sort of effect on cell proliferation. However, we wanted quantitative data as well, so we conducted an MTT assay. And in this assay, we took those treated cells and we added an MTT reagent, which tests for the amount of metabolically active cells that were present. If you turn your attention to the graph on the right, you will notice the x-axis explains the different concentrations. So the light blue on the left is our control, and then it steadily increases in concentration starting from 10 all the way to 80. Our y-axis is the OD values, or in this case, the OD values represent the amount of metabolically active cells that were present. Through the graph, we can see the trend that as the concentration of apigenin steadily increases, there is a decrease in the amount of metabolically active cells that were present. We see the biggest decrease in the 80 micromolar, where there was an 85% decrease in amount of metabolically active cells with respect to our control. The two smallest treatment groups, the 10 micromolar and the 20 micromolar, had very minimal impacts with only 13% decrease and a 30% decrease. We saw the initial jump in decrease at 40 micromolar with a 60% decrease in metabolically active cells. Oh, sorry. Furthermore, our p-value for this assay was less than 0.05, so we were able to determine that it was statistically significant. We also conducted a caspase-3 assay. Now, we did this assay to both supplement our data from the MTT assay as well as to try to figure out what was causing apoptosis. Now, caspases are a very important family of proteases in the apoptotic pathway. Specifically, caspase-3 is known to help with things like DNA fragmentation and also both cell blebbing, which are important processes in apoptosis. In this assay, we took the treated cells and we added a substrate, and if caspase-3 was present, it would cut the substrate and create a color change. So if you turn your attention to the graph to the right, you will notice once again the x-axis shows the amount, the different concentrations of apigenin treatment, and the OD values in this graph represent the amount of caspase-3 that is being produced. Once again, I'd like to reiterate that if there is an increase in caspase-3 production, it means that more apoptosis is occurring. So if you look once again at this graph, the 80 micromolar shows that it had the highest impact with the over 100% increase in caspase-3 production with respect to our control. The two, once again, like our MTT assay, the two lowest concentrations, 10 micromolar and 20 micromolar, had very minimal impacts. However, the higher concentrations did effectively improve apoptosis. Once again, our p-value was less than 0.05, therefore our results were statistically significant. We also wanted to do a flow cytometry test. Now, in this test, we were testing for the expression of PD-L1. Now, PD-L1 is a transmembrane protein that is expressed in a lot of healthy cells. When healthy cells express PD-L1, they bind to the PD-1 receptor on T cells. And now this indicates to the T cells that they are in fact healthy and prevents the T cells from destroying the healthy cells. Unfortunately, cancer cells tend to be a little tricky, and now they have started expressing PD-L1 as a way to evade our immune system. So when cancer cells express PD-L1, they'll bind to the PD-1 receptor just as normal cells. However, this time, the T cells are confused and do not identify the cancer cells as cancerous. Therefore, they're allowed to proliferate in our bodies. So we conducted a flow cytometry test to determine the expression of PD-L1, and there was fluorescence added to a PD-L1 antibody, and then this was placed in, the antibody was added to our treated cells, and then these were placed into a flow cytometer. And in the flow cytometer, there's a laser light that is shown on an individual cells that can detect fluorescence. If you turn your attention to the graph on the right, once again, the x-axis actually shows the amount of fluorescence that is available. And the y-axis shows the amount of cells that we incubated. So in This graph, the more the graph moves to the left, it means that there is a decrease in expression of PD-L1. So once again, we only tested the 4 highest concentrations because we knew these were the most effective. The 100 micromolar concentration of treatment had the biggest impact with a 70% decrease in PD-L1 expression. However, unlike our MTT assay and the caspase assay, the 40 micromolar treatment did not have much impact on the PD-L1 expression with only a 10% decrease. We also wanted to see how apigenin would affect the normal cells in our body. So we looked at a cell line called HEK293 T cells. Now these are a human embryonic kidney cell line, and they're just supposed to model the normal cells in our body. So the same procedure that was used for the MDA-MB231 cells were used. However, these cells were cultured in a different media, and also they were placed in a CO2 incubator. Through this graph, our OD values once again indicate the amount of metabolically active cells that were present. However, you will notice that our p-value for this assay was higher than 0.05. Therefore, any of the small changes that you'll see within the concentration groups were proven to be not statistically significant. Therefore, we were able to conclude that apigenin would have minimal harm to the normal cells in our body. So to go to our conclusion, our 3 hypotheses that we went over in the beginning of the presentations was able to be proved. Apigenin decreased cell proliferation in a dose-dependent manner, and we were able to prove this through our microscopic results, where we saw the morphological changes, and through our MTT assay, where we saw a decrease in the amount of metabolically active cells that were present, and in our caspase assay, when we saw the production of caspases being increased. We also were able to determine that apigenin is effective in decreasing PD-L1 expression, and we were able to prove this through our flow cytometry test, in which we saw the fluorescence being decreased. We were also able to determine that apigenin would have no effect on the normal cells in our body, and this was through our HEK293T MTT assay, where our p-value was higher than 0.05. Therefore, any changes that we saw were not statistically significant. So to move on to some future directions of this research, it would be beneficial to also do some in vivo studies just to see if there are any neutralizing factors or even confounding variables that cannot be accounted for in our in vitro studies. It could also be beneficial to do some drug-targeted therapies for apigenin just in case there are some cytotoxic effects that we were not able to witness. The most logical next step would be to subject the HEK293 T cells to some PD-L1 and caspase-3 assays just to see if that there is any apoptosis occurring or if there is anything that could harm the cells. However, from our research, we were able to conclude that apigenin had the potential to be a really good anti-carcinogenic agent. I would like to thank especially Dr. Gaikwad for helping me with this project. He took me under his wings and gave me the opportunity to conduct this research, as well as Dr. Iyer from NSU. She let us use her flow cytometry machine there and also helped us analyze some of the data points, and also to the biology and chemistry department for both supporting us and also funding this project. Thank you. Questions? Dr. Rainier?
That was a great talk.
Thank you.
That was really good. Apigenin, what did you solubilize it in?
DSMO.
Okay.
Yeah.
So your control was DSMO alone.
Yes. Oh, no, sorry. It was just the cells. I mean, the cancer cells. Yeah.
So apigenin is a powder?
Yes. You can get it, I think, as a liquid or a powder. We used a powder. Yeah. Oh, yes.
What exactly is statistically significant?
It means that any variations that you saw were not due to chance. So if your p-value is higher than 0.05, it means that any variations that you have were maybe due to chance or random probability of that occurring. Sophia?
How much like a accelerated spinach intake would it be?
So unfortunately, these sources have very little apigenin. I think the highest that I saw was chamomile extract, which had 1.2% apigenin. So you'd have to have pretty concentrated amounts. which is something that we could work towards in the future. However, just having like chamomile tea or like adding parsley could benefit your bodies.
Thank you very much.
Thank you.